Hello, and welcome to the second part of our lecture on the scientific method. In this part, we will zoom into the role of statistics in the scientific method and how scientists use statistics to objectively analyze their data, but also create hypotheses and create experiments. In the previous part of the lecture, we discussed that we have a certain hypothesis and we design an experiment and we make a prediction on what the result of that experiment might look like if our hypothesis is true. Now, under many circumstances, and especially when you do statistical research, it's not always clear that there is a difference between hypotheses and prediction, and actually there isn't actually a difference. Sometimes our prediction or our hypotheses are one and the same thing, and if this happens, we talk about what we call a statistical hypothesis. And when you have statistical hypotheses, you do the same thing in the scientific method. You create an experiment, you do the experiment, you statistically analyze the data, and then you compare the prediction or the hypothesis or the result to the hypothesis, which is actually the prediction, and you see that, okay, is it correct? Okay, what does this mean for building our hypotheses and using our hypotheses to build new theories? Or should we revise our hypotheses? Was our understanding of the phenomenon we're investigating wrong? Now, in statistics, we automatically have two sets of hypotheses that are always fixed and that are mutually exclusive. On the one hand, we have our null hypotheses. We try to look at a relationship between a dependent and an independent variable, and we try to check if there's an association. Now, our null hypothesis is always that there is no association between the dependent and the independent variable. The independent variable is the variable that you vary yourself, or that is varied within society. For instance, the more people you meet, the more the higher the risk for getting corona, right? So the independent variable is then the more people you meet because you vary that, and it just varies because of people interacting more with each other. The dependent variable depends on the other variable. So the chance will depend or not depend on you varying the independent variable. The alternative hypothesis is that there is an association between the dependent and the independent variable, and more importantly, that this association, this relationship is statistical. So it's statistically significant and not just due to random error. Now, like I said, they are mutually exclusive because if we reject the null hypothesis, we accept our alternative hypotheses. Now, if we look at an example for this, you can have two variables. For instance, we have a certain plant and we want to see what the effect of giving the plant more or less water each day is and how that affects growth. In our experiment, the null hypothesis will be that the number of plants that survive, or how fast they grow, is not related to how much water they receive. The alternative hypothesis will be there is a correlation. The more water you give to a plant, the more likely it is that the plant survives up until a certain optimum. Of course, when you give more water, nothing will happen anymore. But this correlation can be either positive or negative, but at least there's going to be a statistical correlationship between statistical correlation between your independent and your dependent variable. This can result in four different situations. You can have a situation in reality where the null hypothesis is actually true. So in our experiments, it would mean that there is no relationship between the amount of water you give to a plant and the survival rate of a plant. This can lead to two other situations. It can be that you actually also find in your experiment that the null hypothesis is true. In this case, your experiment will be correct. But it can also be that your null hypothesis is rejected while it should be accepted. This means that you made an error, and this is what we call a type 1 or alpha error. It can also be the other way around. The null hypothesis can be false. Your experiment can indicate that it is actually false, which is correct, or it can indicate that it's true. So there might be a correlation between two variables in real life, and you can still accept the null hypothesis, that being that there is no correlation. So this means that you made a type 2 error, a beta error. How can we avoid these statistical errors? How can we make sure that we don't get these errors, and the result of our experiment actually reflects the situation in reality. Now, it all starts with good experimental design, and therefore this is very important in this course. Obviously, it's very important to construct your experiments in a very well taught systematic way. One thing that is important, you should only have one independent variable, right? You should make sure that the independent variable that you will vary is the only one that influences the dependent variable. Otherwise, it will be difficult to account what type of effect in the dependent variable is due to which independent variable changing. This is obviously not always possible. For instance, in our case, we do biosensor research. So let's say we have an electronic sensor, right? And let's say that we are detecting a certain target, and we know when the target binds to our receptor, this will change the resistance of the receptor. We will measure that as a difference in our electrical signal. But what if we also know that the independent variable can also change the pH, and the pH will obviously also have effect on our electrical signal. What part of our change in signal is then due to the binding, which is what we're interested in, and what is caused by another independent variable? And that is not something we're interested in. So this is very important, a very important example that shows you that sometimes, although your experimental design is perfect, you can still have a problem. Now, what you should always avoid is that your experimental design is the origin of your problem. So what you always should avoid is having bias in your experimental design already. When we think about bias, we usually think about what we see here in the picture. Well, we won't be needing that sample. So a bit of confirmation bias, or bias that is created actually by the people doing the research. So they have a certain opinion, or they want a certain result because that will make them famous, or get them patents, or get them money, and they will just commit fraud. Now, there is a difference between personal bias, bias by the researcher, fraud, and experimental bias. Experimental bias is different in a way that it's just due to bad experimental design and might not be something that a scientist consciously does. Experimental bias can be, and is usually in terms of statistical hypotheses, due to sampling bias. Now, when we measure or when we do an experiment on a certain population, it's often not possible to study the entire population. For instance, we can't study the whole of the Netherlands when we want to know something about COVID or test the efficacy of vaccines. We take a smaller subset of the population and we measure within that group of individuals. We do our therapy and we use a placebo group. So this is the sample of the population. Now, what can happen is that your sample is wrong, that there's sampling bias. And this means that the sample is not representative for the population or inappropriate for the research question you're having. So what this means technically usually is that either your sampling size is too small. When you have a very small group of people, there can be a random effect in that group of people that influences the results. By having a large sample size, you avoid that. More importantly, you should also look at the selection of your samples. How do you do it? Ideally, you do random sampling. You make sure that the composition of your sample matches the composition of your population. You take persons from the population at random, so you have a nice spread between different age groups, different genders, and different, I don't know, whatever can influence your result, BMI, stuff like that. And by doing so, and by taking this into account before you even start the experiment, you minimize the chance of sampling bias influencing your results. Another important form of bias that's associated, second very important form of bias that's associated with doing your experiments, what we call measurement bias. Sometimes you're doing a type of measurement in your experiment to look for a correlation, but the data collection method is not chosen in such a way that there is an ideal match between experimental data and reality. So if that is the case, if you see that something is wrong with your data, and you see that when you study the population as a whole, you see that the results from your experiment don't match what you would expect in reality, you should reevaluate your technique. You should make sure that, am I using the right technique, but aren't we doing the measurements accurately? Make sure that there's a minimal influence of environmental factors. Isolate the effect of multiple factors. factors and do sufficient replications. So again, to get back to the example we had before where we have an electronic sensor that is able to detect a certain target, let's say COVID, what we should do is minimize the effect of um, differences in pH between samples, differences in temperature between both days. And what we usually do is what we do to isolate the effects of multiple factors is have a control, a positive and a negative control. A positive control being a sample where we know we should have a large signal. A negative control is a sample where we know there shouldn't be a signal at all. And we also use a reference channel where there is no receptor layer. So any effect measured in that reference channel should be due to the fact that something changes in the environment. And then by comparing positive and negative control and your reference channel to your actual sample channel, you can subtract the non-specific effects of your environment from the actual effect of COVID, for instance, binding to your receptor. And that's how you can avoid the limits of your data collection method influencing the conclusions you make from your experiments, and that's very important to avoid bias in your results. Now, experimental bias, to summarize, is bias that is not due to the scientists doing it, to the humans interfering with the scientific method, but it's bias that's inherent to the experimental design. And by optimizing our experimental design, we can also avoid bias, and in doing so, we can make predictions on our hypothesis and test if the data matches prediction, and not only what that would mean in terms of reality, but also we also have to think about that data, is it relevant for what we're trying to prove. So that's also something you need to take into account, and it's something that scientists do all the time. So do an experiment, and then, okay, this might mean that our hypothesis is correct, but we need to be critical. It might also mean this or this, so we need to do more experiments and dig deeper to further support or to find further support for hypothesis, and if not, revise our hypothesis and make it more correct, more in estimation with reality. Thank you for your attention.